In this video, we will have a look at 3 different annotation viewers: the Cohort Overview, the Annotation Plotter, and the Cohort Sunburst plotter. These viewers provide an overview of what annotation is available for a dataset, the values that the samples have for a specific annotation, and the respective shares of each value in the dataset. Furthermore, they allow the user to visualize the annotations for manuscripts. In R2, the samples of a dataset can be annotated with extra information, such as clinical data or biological characteristics, and each group of annotated data is called a track in R2. These tracks can be used in most of the analyses in R2. Tracks will allow you, for instance, to filter datasets, to compare groups of samples, to color plots, scatter plots of samples with meta information, or to correlate genomics patterns in your data to, let's say, different phenotypes or demographic characteristics. Sometimes you just want to see which annotation is available for the samples in a dataset. For such occasions, you can look at the annotation plotters, and you can find them under the header annotation. We will have a look at these 3 plotters, and we will start with Cohort Overview. So we click on Cohort Overview and click Next. Before we get into this module, I would like to address a small change that happened in the R2 platform while we were recording this module. The R2 team constantly works in the background to further develop the R2 platform. So new modules are integrated into the platform and old modules are updated for better functioning. That's why you might see slight differences with the video when you go to the platform and follow the tutorial. In this case, further down in the video, you will see the values of this upper track here with the symbols greater than or less than or equal. So both here and in the table, you will find the symbols instead of written out as it is here. Keep in mind that these are the exact same tracks and that you can just follow along with the tutorial. It's the same dataset, same tracks, and it's just a different writing. As I said before, tracks or annotation is extra information about the samples of a dataset. Let's start with the grid at the bottom. Here you see the annotation information that is known for the samples of this dataset. The first column shows you the sample names. Then you see which other kind of annotated information is available for this dataset from the headers of the columns. Following sample ITCC 0013, we can see that the row colors gray when we hover over with the mouse over this sample. We can follow each of the columns in this row to see more characteristics of this sample. For instance, it was annotated as stage 4 of the INS track. We can see that the age year is 2 and that the sample was annotated as female, and many other characteristics. You can see that tracks can be numerical, like the age year, or categorical, such as gender, female or male. This information can be used in many of our analyses. Whenever you want to analyze a subgroup of your dataset, or when you want to color a plot, you can use a track. Also, you can use track in the Kaplan-Meier analysis, for instance, to find the difference in survival chances for high-stage neuroblastoma versus low-stage neuroblastoma. And you can find videos about how to do that in our playlist. In this cohort overview, you furthermore see some pie charts on top of the page. You can adapt which pie charts are shown. And also over here, you can adapt the pie charts. If you click on any of the shares of a pie chart of any track, the table below is adapted to that selection. So for instance, in this case, we selected from the track age group the samples that show the value more than 18 months. And we can see that here in the table underneath, only samples are now displayed that have the value more than 18 months. Once you have selected a specific group, such as now we have both made a filter of more than 18 months and male, and we are done with this overview, we can say clear filters to go back to the default table overview. At this point in the video, I actually inserted 2 more functionalities of this cohort overview. that are recorded a bit later. Therefore, you can see that the age group track, here the first column of the grid, but also here above, has the adapted textual description instead of the more than and less than symbols that this track was described with earlier on. Anyway, it's the same track, same values, only textually written out. So let's carry on with the functionalities that I wanted to show. So in this grid, you find in the headers of each column the option to filter the samples. If you click on these horizontal lines, then you find a little menu to sort, but also to select a certain value of the track. And you can say filter. And you can see now I filtered, and both the pie chart and the table, they have adapted to my filtering options. I can also undo this again by either clicking clear or just rechecking the other value and say filter again, and we are back with the normal view. This can be done with any of these tracks, and you have different filtering options for categorical than for numerical tracks. As you can see here, you also have these kind of logical filtering options, less than, where you can, in the drop-down, find other kind of logical options. Another feature of the cohort overview is the Build a Track button over here, underneath the grid. You can click on it, and you will be asked based on which available annotations you want to make a new track. This means that you will be able to combine the values of different tracks into one, and this new track will be available to you for all the other analysis with this dataset as well. So as an example, we could choose the MIC-N amplification, and let's choose gender. If we are happy with our selection, we click on OK. And you can see that now every sample here is listed with its value, which is a combined value from both the MIC-N amplification and the gender. Over here, underneath the samples, you can choose a color, and also you can adapt the labels. So we could add MIC-N and add MIC-N to every label for us to have a more clear idea about the values. We can also rename the track. This is MIC-N amp underscore gender. And we can choose to show the track by default underneath any plot. And we can also choose whether we want to store it for 24 hours or that we want it to be available for us until we delete it from our personal settings. So this means that if we leave it for 24 hours, it will automatically be deleted after 24 hours. But until then, we can do any kind of analysis that involves tracks. So then we click Build Set, and it has been updated. We can both see this if we go back to the cohort overview. You can see now that both in the table over here, you can see the track temporary T, MIC-N amp gender, and you see the values MIC-N no female, MIC-N no male, etc. Also, you could just show in the pie chart and look for the T MIC-N amp gender to show which shares every value has. And we can see that the non-MIC-N amplified male is the largest share. Furthermore, we can find back to manage under tracks in user options. And since this is a temporary track, we find it back under manage temp tracks for this data set. And indeed, here we see our track name, where we can again adapt it, or we can export it. But also just to show that this track now is available throughout the analysis, we can do, for instance, a fine differential expression between groups and select our just defined track and click the MIC-N non-amplified male versus the MIC-N amplified male. And we click next. And we now get a list back of the genes that are the most significantly differentially expressed genes between the two groups, non-MIC-N amplified males and the MIC-N amplified males. Another more simple track viewer that is often used for publications is the Annotation Plotter. Again, you can find it in book 3 under the Annotation modules. Here you find the Annotation Plotter and you can click Next. In some publications, patient data is represented in slick-looking annotation plots, showing the patient characteristics in rectangles. In a sense, these are just like the tracks that see in R2 as well under many of the plots, mostly YY plots. To allow users to create these track figures ordered in a user-provided order, we have implemented the Annotation Plotter in R2. The default view for the dataset will be plotted. If you want to plot other tracks as well in this view, you can simply go over to the Track Display selection, click on the arrow before default, and check any other track that you are interested in, such as age year. If you then press Adjust Settings, you can see that age year now is displayed as well. Furthermore, you can change the track sort order. If you would like to sort the samples by gender, for instance, we simply drag over gender to this area and we say again Adjust Settings. Here we see that sorted by is now on gender, and we can see that the first gender is female, the second gender is male, and the tracks above are adapted in order. That's all for now about the Annotation Plotter. Let's go back to the main page. As you may know, the R2 platform is under constant development. Whenever we hear about a useful analysis type, we try to add this new analysis to R2 such that our users can analyze their data with this new type. Since our last recordings, we became aware of the Sunburst annotation plot. And now you can find it in book 3 under Annotation Viewers as well. So let's have a look. Over here under Annotation, you can now find also Cohort Sunburst Plotter, and you can just click on it, click Next. And then in this next page, you can decide which tracks you are interested to see and in which order. So for us, let's start with staging information, INS staging information. Then I would like to see the MIC-N amplification information. And let's end with the live track. And then you can just click Adjust Settings. I just want to note there are also other plot types that also have a hierarchical structure available in here. But for now we will stick with the Sunburst. And click on Adjust Settings. As you can see, the Sunburst annotation plot has unfolded, and you can see that the 3 tracks are displayed in a circular hierarchical structure, and that you can follow certain characteristics to find out the amount of samples that are in that subgroup of the dataset. So for instance, here you see stage 4 as one of the possible values of the INSS staging information, and the dataset contains 40 stage 4 samples. Then to the second ring, which has the MIC-N amplification information, we can see that in the stage 4 group of 40 samples, 15 of the samples were annotated as having indeed a MIC-N amplification, whereas 25 did not have this amplification. Of the 15 samples with stage 4 and a MIC-N amplification, 13 were annotated as not being alive and 2 were annotated as alive. On the other hand, we can see that in the group of stage 4S samples, the stage 1 samples and stage 2 samples, no MIC-N amplified samples are found. And also we can see that in the stage 3 there's 1 sample and this sample also was annotated as not alive. Overall, we can see that in stage 4S, stage 1, stage 2, and stage 3, most of the samples are annotated as alive. You can click in the plot to adapt the plot to only a subgroup of your data set. For instance, stage 4, you would click on stage 4 cell and it would only show in the circle the samples of stage 4 with their values for the tracks that we have chosen. But also you can still adapt the tracks that you have chosen simply by dragging the tracks in a different order or completely exchanging them for other tracks. For instance, if I remove the alive track from the right side and I import the age group to the right side and click adjust settings, we see that the Sunburst plots adapt to this new track. Don't forget to also check out the other hierarchical annotation plotters, but for now we leave it to this and go to main. To know more about the statistical relations between tracks, we advise you to watch the video on annotation analysis that you also can find in the playlist.